and thank you for the invitation. It's been very interesting until now. I hope I can convince you that that the immunology of allergy vaccines is also very interesting, or quite interesting. I'm from a medical company producing allergy vaccines, so that's kind of a disclaimer, and I'm not altogether agreeing with David Raitt that vaccines based on full-length proteins doesn't work. We have been doing that for almost 100 years, and we are pretty happy about it. So what I would like to look into today is what is allergy immunotherapy all about? Just touch upon what are we actually doing with our vaccines. I will rush you through some cellular markers and get to the actual focus of the talk, the antibody-mediated changes, because that's what we what we try to link to the clinical effect, and that's what we are also using in our communication with the authorities when we develop new vaccines, and we use it for dose finding, long-term effect, and also how many species of different allergens do we cover with the individual vaccines. We have two different, we have a number of different approaches to do allergen immunotherapy. The most known ones, or what we focus on nowadays, is the subcutaneous immunotherapy, subcutaneous down here, and the sublingual immunotherapy. And for the sublingual, we have developed tablets recently for the big allergies like grass and ragweed and house dust mite. Much of what we know of the mechanisms is from studies done on subcutaneous immunotherapy, and I'll show you details from that in a moment. Of course, there's also a lot of what we call experimental immunotherapy, new ways of doing it with different different routes of administration, and also, as David Raitt just mentioned, the studies from the Circassia based on peptides is also coming up recently. What's going on when we do immunotherapy, and why do we think that immunological changes are important? Of course, it's a vaccine, and all of us would agree that vaccines do something to the immune system, but we also see long-term effects in this vaccine approach, so that's why we would like to look into that. This is a characteristic profile of a well-performed trial on allergen immunotherapy. You have the pollen season here, it's grass pollen treatment, and then you register symptoms, and medication use, and also ask the patients how do they feel compared to the last season they did not have this treatment. And as you can see, comparing the immunotherapy with the placebo group, there's a big difference between this, and this is highly significant on all parameters that you measure. The important thing with respect to the immunological effect is that when you go into some years after treatment and take the same patients and split them into some that continue on immunotherapy and some that stops, the red ones, you can't really distinguish between the two groups. So there has been some kind of change induced in the immune response that account for this difference compared to it's a new control group, but it's still untreated patients that are compared to. But the interesting thing is that you can't distinguish between the one that continue on treatment and the ones that doesn't. So something has happened. The current model for this is, well, if we start out with what happens when you meet an allergen, when you are sensitized and respond to it with an allergic response, you have a Th2 response, you have the production of IgE, the IgE binds to the surface of the mast cell and the basophil, and you get your allergic reaction, and at the same time the T cells produce IL-5, and you get your eosinophils, your development and influx of eosinophils into the target organ, and you get the allergic response. Then when you do this, and this is a model from Robertson, Larcher, and Durham, who's been very much active in this field, they represent the treatment as in the green lines here, where you have high dose and you have a different route of administration. You get the Treg induction, you get also Th1 induction, and the regulatory cells down regulate the Th2 response primarily, and then you get a different profile of cytokines produced, and you get the antibody responses changed. And what has been much focused on lately is the regulatory cells. Everybody are talking about regulatory cells, so that I will go through to start with. Also, the shift from Th2 to Th1 response has been described. We have done a lot on the antibody responses, and that's where I will end up my talk. So, starting out with the T cells, Chesmi Actis and Kurt Blaser published this very key paper back in the late 90s, where you can see that the production of IL-10 was their key finding in this paper. You have the downregulation of all the Th2 cytokines and the proliferation, and actually also of interferon gamma. So the Th1 response is also downregulated initially, but the key finding was the induction of this IL-10, and that has been a dogma since then. This is supportive information that you can see if you, after 60 days, you have this very clear reduction, and if you include an anti-IL-10 antibody in the experiments, you kind of restore most of the reactivity. So clearly IL-10 is important for the effect of immunotherapy, allergen immunotherapy. We've been looking into the tablet treatments and also found that that if you define a regulatory phenotype of T cells, this is in an unspecific system, so we just look at the number of regulatory cells defined by these markers, or these markers where it's the CD25 high we zoom in on. You can see that we see some increase in the number of regulatory cells, but as you also can see, it's quite small increases. The number of cells, or the percentage of cells, increase from 3 to 4%, or in this case from 1 to less than 2%. But clearly significant, and being a basis of the explanation of the mechanisms behind the treatment. Eric Wamper published a couple of years ago a very nice study based on MHC class II tetramers for grass allergens. You can see that in this case you have the tetramer positive up here, and you have different cytokines down here, and this is the allergic patient with a large proportion of Th2 cells, IL-4 producing, and the other one over here is the non-allergic patient, a good example of that, with production of interferon gamma and also some IL-10 production. IL-17 is very little or almost not present in these patients. If you look at such profiles, and I think it's 12 or 15 patients involved in this study, you can see for different allergen epitopes, you have a profile in the grass allergic patients that is very much dominated by Th2 cells, it should say out here, at least for the majority of the epitopes. If you go for the non-allergic patients, you have a dominance by the Th1 cells. I'm sorry I missed some of the slide here, but it's clear that they have a very much different profile than the Th2 cells of the allergic patients. Of course, you have less number of cells here, so maybe there's a bigger variance on the actual numbers, but there's clearly a difference between the two profiles. And then, if you look at patients after treatment, you can see that they have shifted from the allergic profile up here to something that more resembles the Th1 or T regulatory, which are the blue ones, profiles of after treatment patients, or also seen in the non-allergic patients. we did something similar. In this case, you're zooming in on the individual T cell epitopes in a collaboration with Alessandro Sette and his group in La Jolla. We zoomed in with peptide pools in an ELISpot assay. We looked for T cell responses to the individual peptides. A combination of around 20 peptides was used to detect T cell responses in ELISpot. This is IL-5 and IL-10, and as you can see, you get a down regulation of the IL-5 response, both in subcutaneous and in sublingual, even though it seems to be less in the sublingual treated patients. On the other hand, you have a clear induction of IL-10, but transient induction of IL-10, in this case, both in subcutaneous and in sublingual immunotherapy, but primary in the sublingually treated patients. So there's also people looking into the innate immune system, looking for differences between treated and non-treated patients, and this is just to mention that there are examples of DC markers that are related to tolerance as such, which is seen to be upregulated in treated patients. And also recently, Mohammed Shamji and his colleagues, Stephen Durham's group, looked into the number of ILC2s, which could be kind of an amplifier of the allergic response because they produce the same Th2 cytokines as the Th2 cells, the specific response. For this, looking into or using this in the explanation of the mechanisms behind the treatment and in the communication with the regulators, it might be a little difficult to explain an increase in a gene expression of markers that to some extent maybe is not so known for their regulatory properties, or to look at downstream effects of these ILC2 cells, so we're not really looking into that currently. But this could lead to a very much more detailed model for what is going on. But I will just say that now I will switch to the antibody production and look for what kind of changes do we see in that part of the immune system. So this is subcutaneous immunotherapy, and you have a 12-week treatment period here. So it's very early. It's in the updosing phase as you use for this kind of treatment. But you can see that this very clear increase, in this case you measure IgG4, but other antibodies would show the same profile, even though IgG4 is the most clear increase that we see. But at the same time, you also see an increase in IgE. So what's going on? I just tried to convince you that it's the Th2 cells that have been lost, and the Th2 response should be down-regulated. But even though we see IgE responses either going up or are unchanged. So we expected that it must be something with the interaction between these two responses that is accounting for the effect of immunotherapy on the antibody level. And looking into that, we look at the function of the immunotherapy-induced antibodies and what happened to this non-IgE that is induced during treatment. And we can do that in a binding or blocking assay. So we catch with a solid phase a particle that has anti-IgE on the surface. We catch the IgE, and we do that in two different ways. One, where we keep or we wash away all the other antibodies present, or we do it in a situation where we don't wash away, and it looks like this. So wash on this side and no wash on that side. It's pretty simple. You get rid of everything that can compete with the IgE for allergen binding. And then when you add in the allergen for detecting how much binding you get, then you get a different situation in these two assays or assay conditions, and then you have the balance or the ratio between these two systems being when you have blocking antibodies present, you get a reduction of the IgE binding. The next thing we could do is doing a basophil activation test, and that's more straightforward, directly related to the reactivity of an allergy or an allergy-specific cell in the blood, the basophil. We use strip basophils depleted for IgE on the surface. We add the post or the pre or the post immunotherapy serum. Then we have these two situations again, where we have either with the blocking or without the blocking antibody and with the blocking antibody. And then we add the allergen, and we only get release when we have the situation where there's no competition, or at least we get a reduction of the release in the situation down here. The final one is what we call the FAB assay, which is facilitated allergen presentation. That's based on, we have a B cell in culture, and the B cells are expressing CD23. And this has been shown to be enhancing or facilitating the presentation of the allergens when you have IgE present. So when you add the IgE from the serum and you add the allergen, then you get complex formation on the surface of the B cell. And this is taken up and presented to the T cell, and then you can have a readout where you detect the antibodies, the IgE antibodies bound to the surface directly in a FACS analysis, or you can have the T cell activation as your readout. But in both cases, if you add in serum where you have the blocking antibodies present, then you will get a reduction of the T cell proliferation or a reduction of the binding of the complexes to the surface. And when you do that and investigate post-immunotherapy sera, then you get a situation like this. I didn't show the placebo-treated patients because they are completely flatliners. You don't see any kind of effect on the placebo patients. So IgE binding, you have a reduction after the first year of treatment. This is Birch immunotherapy, 2 years of treatment, and it's maintained during the second year. And the same goes for basophil, maybe slightly delayed in the basophil readout, but still you have a strong reduction and clearly significant. And the same goes for this surrogate for T cell activation. So the antibodies are really playing a role. And what's going on if we look at the stoichiometry of the IgE versus the IgG? In this case, we have two different specificities of IgE present to make the complexes. Down here, two high affinity IgE antibodies. We made a panel of IgE antibodies to investigate this. And we can switch the isotype of the antibodies, so we have the versions of both IgE and IgG of the same specificity. So if you add in 1 to 1, 1 to 5, you have to go to 1 to 10 to get a complete blocking of this complex formation. And that's even in a situation where you have a complete match between the IgE and the IgG. So our thinking is that you need quite a large excess of IgG to have complete inhibition of these reactions. And when we compare the histamine or the basophil activation test, the facilitated allergen presentation, and the IgE inhibition assay, then you can see that we have quite nice correlations for all three of them. So we believe that it's the same mechanisms that are in play, the same stoichiometry is needed to compete in the different situations. By the way, when I say inhibition or binding, it's just which way the curves are going. If they're going down, then it's binding. If they are going up, then it's inhibition we are measuring. Doesn't make much of a difference. Okay, but what we really want to do is also, of course, correlate this to clinical effect. And to do that, we went to a study where we had treated around 400 patients, but we had samples for 200 of those. And we have treated for grass allergy, we have treated with a very high dose and with a non-optimal dose of grass pollen, and we had the placebo group. And as you can see here, whatever measure we do, in this case we also included a direct measurement of the IgG4. However we do it, we see a very nice difference between the actively treated and placebo. We see a time dependency, and we also see a dose dependency where the 100,000 group is very much better to induce these antibodies than the 10,000 group. So we also would like to correlate that to the clinical effect, and this is the combined symptom and medication score that normally is used used in registration trials. So that is what the authorities want to see, I completely acknowledge that this is not the best of correlations. The R values are not very big, but at least we find significant correlations for 2 of the 3 parameters, the binding or blocking of this to the B cell surface and the IgE blocking factor, as we call it. For a direct measurement of the titer, the IgG4 titer, we didn't find a correlation. The important thing here is that this is just maybe a simple representation of the data. So when we do a modeling of the data, and when we do exclude the placebo patients, we can see that we find the same kind of correlation. So we're pretty convinced that we have a significant and also a relevant correlation in these data. We could also show that we have a big part of the variation of the data is accounted for by the immunology readouts. And when we normalize the data based on the immunological effects, we can show that we lose the difference between the groups. And all of this is on the way towards validation of a biomarker for clinical effect. But of course, with the still starting out with a correlation like this, you don't have a full validation of a surrogate parameter. However, we find similar correlations in other studies. In this case, it's steroid use that is the clinical readout. In this case, it's direct just allergy symptoms. And also in a small study we did with combining both sublingual and subcutaneous immunotherapy, we can see that we have a nice correlation between the seasonal symptoms and the basophil activation. To expand on this, we also try to include a nasal allergen challenge because the variation on the clinical readouts here is very big. So we often have problems even seeing differences between the different groups on that parameter. So correlating that to an immunological parameter is very difficult. So we would like to have something that is more clear-cut for the clinical readouts. And as you can see here, we have a nice correlation between the seasonal symptom scores and the nasal challenge. So we might also convince the authorities that at some point we can use these clinical or the challenges either in a chamber or direct allergy application to the nose as a readout for these kind of trials. Okay, the tablet treatments that we have recently developed for grass, we can see the antibody-derived effects here, and you can see that over the years with the placebo group, no change at all, and you have this clear induction during treatment. Treatment stops at this dotted line, and then you have a slight reduction, but still a very highly significant difference between the 2 groups. And that goes for either the blocking factor, the G4, or the FAB, the facilitated allergen presentation, the T cell readout over here. When we do that in various trials where we have done data from 5 big trials, this is a number of years included from one big trial where we did 5 years treatment, and then other trials where it's 1 year of treatment. You can see that there is a correlation between the key or the primary readout, the difference in the primary readout for the trial, and then the immunology. In this case, it's G4 we have used as the indicator. Maybe the most important thing is that one study that did not meet the primary endpoint also have a very bad or low induction of the immunological change that we normally follow. So, Carisax is our mite immunotherapy tablet and the same immunology on G4. And I found out it's much easier when you draw the curves yourself, then they look much more nice. So going to the lower dose, the medium dose, and the high dose, you can see that there's a clear both time and dose dependency in this case, and that goes for both of the species. These are the two mites that are the most important inducers of mite allergy. So you can see that you have a nice response to both of them, and they are both included in the treatment. The immunology, also G4, if you look at the clinical effect in a study where we, instead of going to the 6 DU before, we went to 12 DU, you can see you can't really distinguish on the clinical readouts between the two higher doses. And that also reflected in the immunology, where the two high doses here are very much alike. On the other hand, when you look for the blocking factor, you might have a little better resolution. So again, you have the nice dose and time dependency when you look for in the lower doses. But when you go for the very high doses, the 6 and the 12 DU, compared to placebo, then you have actually a significant difference that could suggest that you might boost your immune system a little bit more, even though you're not getting a full representation of this in the clinical readouts. And also something we are really happy about is that when we use these markers to also communicate with the authorities, that we see the same profiles in Europe, US, and Japan. So maybe we will get off a little easier when we are introducing these tablets on new markets, if we can show that we have the same immunology in Russia and Asia, wherever we want to put this in. Finally, I'll show a little how we use also the immunology to show that we have species coverage. When we talk about different treatments for ragweed immunotherapy, this is 3 different ragweed plants, and in the States they are very keen on treating with exactly the plant that they have diagnosed the patients with. So we have these 3 that are important, and some doctors like this one, and others like this one, and the third group like this one. So we are up against some communication issues on what's the best way to treat. Our tablets are based on this species down here, which is short ragweed. And when you compare this to this on the immunology, IgE reactivity, you can see that you have a complete correlation between the two. And the same goes for this one and this one. And as you can see here, the vast majority of the patients are responding to both of them, and equally, as you see with the correlations. When you then look into the G4 responses towards these species, the 3 species down here, you can also see that the G4 is clearly cross-reactive, and you have the same level of response to the different species. So again, we actually tried to use this to convince the authorities that this was enough to lose the short ragweed in the description of how to use this vaccine, but they turned back and said they needed clinical data to show this. I'm really stunned that you can say that this is not a complete evidence that you have this cross-reactivity, but time will show. We will do the clinical data also. Yes, and I hope I convinced you that there's a lot of interesting findings in the T-cell compartment that might at some point be also biomarkers of allergic disease or allergic responses to allergy vaccines. But for now, I think we have the antibodies that are a nice and very consistent finding. We have some correlation to clinical effect. I'm the first to acknowledge that it's not optimal, but at least we can use it in the registration process to look for dose finding and for to parallel studies between geographical areas. We can convince the authorities that if we see the same profile, we will also think that we have the same long-term effect of the treatment, and we can for sure show the dose and time dependency of these vaccines. Thanks to the collaborators, especially Stephen Durham and his group has been very much involved in this, and also other clinical sites has been very helpful. And then thanks to my colleagues, especially Joost van Nerven, who is not at ALK anymore, but has been the one starting up all of this, or much of this, and Henna Gibson, who is a very skilled antibody guy, and our statistician, Christian Løing, and they have been the driving force behind all of this, and of course the whole of ALK research and the ALK in general has been very supportive. Thank you.